Welcome to the session. Here we are going to discuss about the topic egg cortex and its development. Egg is the female gamete which helps in the formation of individuals. So it has to be protected and it consists of a different coating around it which helps to protect it. It's called egg cortex. So we are going to discuss about the components of egg cortex, how it is formed and its development. So, egg cortex. The egg cytoplasm is divided into two layers, surface layer and inner layer. The inner mass of cytoplasm is liquid in nature and is called the endoplasm, and it contains yolk, nucleus, mitochondria, Golgi bodies, endoplasmic reticulum, and all of the cell components, so on. And the surface layer of the egg is called egg cortex or ectoplasm. It is more viscous and gelatinous, and thickness of the cortex varies from species to species. Cortex contains granular substances called cortical granules. They are not found in the eggs of all species. Some do not possess them. They are especially found in few annelids, lamellibranchs, sea urchins, fishes, frogs, etc. And they are absent in insects, gastropods, urodeles, etc. So, the cortex remains thicker around the animal pole and thinner towards the vegetal pole. The cortical granules, small rounded substances, they are arranged near the plasma membrane of the egg, and it contains mucopolysaccharides and proteases, and it is formed from the Golgi complex or Golgi bodies. Egg cortex. So let's now move on to endoplasm, the inner layer. Internal portion of the ooplasm is called endoplasm, and it remains in a physical state of suspension with a continuous liquid phase and large inclusions such as mitochondria and ribosomes dispersed in it. It consists of a cup-shaped mass of white yolk platelets called vitelline cupola at the vegetal pole, and it remains opened towards the animal pole. The yolk platelets are large near the vegetal pole and less densely packed towards the animal pole. Thus, accordingly, a polar axis, an anterior dorsal axis, the animal vegetal axis, which exhibits a concentration gradient from one pole to the next pole, that is, opposite poles. Next is an important phenomena that is called cortical rotation. Egg membrane formation is intimately tied to changes in the egg cortex. Within seconds after the membranes contact with each other, changes occurred in the egg cortex. That is, the egg decrease in size or volume. It gets shrink to a certain size. The dense granular layer of the cortex gets converted to a region of microfilaments adjacent to the plasma membrane. The cortical reaction culminates in the formation or leads to the formation of fertilization membrane outside the egg plasma membrane. So, what's the role of this fertilization membrane? It blocks the entrance of the late arriving spermatozoa, or it prevents polyspermy. Now, let's move on to an example how this fertilization membrane is formed in sea urchins, in the case of sea urchins. The first step, as soon as the apical end of the acrosomal tubule of the sperm touches the surface of the egg, from the site of contact, a wave-like color changes from yellow to white, travels rapidly around the egg cortex, and is shortly followed by the elevation of fertilization cone from the surface and the formation of fertilization membrane. So that's how fertilization membrane is formed in sea urchins. Sea urchin has two membranes, outer 30 angstrom thick vitelline membrane and inner 60 angstrom thick plasma membrane. Two different membranes will be present, and they are formed by three different stages. The outer vitelline membrane, separated from the plasma membrane, undergoes expansion and becomes the outer layer of the fertilization membrane. The cortical granules explode. That's the first one. Second, cortical granules, the rounded structures present in between the plasma membrane, explode, and they release mainly three components. First one, dark, denser, lamellar and folded parts of the granules unfold and fuse with the vitelline membrane. In the second case, globules. Globules fuse together and build up a viscous hyaline layer at the outer side of the plasma membrane. Next, a liquefied component fills the perivitelline space. These are the three substances or products formed from the after the rupture of the cortical granules. And all these structures, vitelline membrane plus the contents of the cortical granules together forms the fertilization membrane. So here we can see this, the rounded structures are called cortical granules. They rupture and forms the, here in the second stage, bursting of cortical granule takes place, forms the vitelline membrane, and vitelline membrane is formed, and later it leads to the formation of fertilization membrane. This is the perivitelline space. And the next, as you can see, the globules fuse together and builds up, say, hyaline layer around it, and that's how a fertilization membrane is formed. This is the egg cortex. Next, how fertilization membrane formation occurs in the case of vertebrates. Cortical granules are broken down after sperm's penetration into the egg cytoplasm in case of vertebrates, and the contents gradually fills up the perivitelline space in between the chorion and plasma membrane in case of bony fishes. In the case of frogs, the perivitelline space is filled in between the vitelline membrane and the plasma membrane. In mammals, granules burst open and release their contents into the perivitelline space that is between plasma membrane and the zona pellucida. So this is a representation of how polyspermy is inhibited. Once the acrosomal tubule touches the fertilization membrane of the egg cell, it induces changes in the egg, and only one sperm will be able to penetrate into the egg cell or to form zygote. The other one will not be able to fertilize the membrane. It goes, or it moves back. Acrosome of the sperm sends a message, and it initiates further actions and prevents polyspermy. So fertilization membrane plays a major role in the prevention of polyspermy, and in previous cases we have seen how it takes place, how cortical granules burst and forms the membrane in between the zona pellucida and perivitelline space. So this is how these are the basic components of egg cortex and how development occurs. Thank you.